Hello, this is Haonan Zhu, and I'm a postdoc at Lawrence Livermore National Laboratory. I'm here to present our work on deep active learning for genetic interaction experimental design, and this work is done in collaborations with UCSF. One of the most effective methods to hinder viral infection is to directly modulate the host gene expressions. And the overall goal of this project is to identify host gene pairs whose combined downregulation suppresses viral replications. However, a naive exhaust search of this combinatorial space is simply invisible due to limited experimental budget. In this work, we develop a deep active learning framework to predict viral loads from sequential gene knockdown experiments. To properly address the budget constraint, we incorporate pre-existing biomedical knowledge graph into our active learning framework. Our model is able to balance exploration and exploitation to more efficiently identify the top-performing gene pairs in HIV genetic interaction data, which demonstrate its promises to develop into resilient antiviral therapeutics. The first figure is a visualization of the knowledge graph we focus on in this study, which is referred to as SPOKE, stands for Scalable Precision Medicine Oriented Knowledge Engine. It's a database of databases, where nodes and relationship properties are drawn from genes, gene ontology, transcriptional profiles from various databases. In total, the nodes of the graph contains over 20,000 human genes, over 200,000 human proteins, combined over 20,000 biological processes, cellular components, pathways, and molecule function entities from gene ontology. In addition, it contains 1 million edges capturing transcriptional profiles, including overexpression, knockdown, knockout impacts. We aim to incorporate information from the SPOKE knowledge graph to reduce the computation complexity of the gene pair experimental design problem. In the middle top is a visualization of the HIV genetic interaction data we primarily use to examine our method. It's a data set collected by the Colgan lab at UCSF, where they handpicked 356 genes that are known to be relevant for viral replications of the HIV viruses, and each entry of the matrix corresponding to a combined downregulated gene pair, and the value of the entry is the measured viral loads. This entire process is labor and cost intensive, and we aim to demonstrate utility of our method by limiting our algorithm to only query 200 entries in the matrix per step, and limiting ourselves to total 30 steps, which corresponding to observing 10% of the matrix entries in an adaptive manner. We want to show that we can uncover the most top for performing gene pairs within this limited experimental budget. In the next figure, we summarize our deep active learning framework using a flowchart. At the first step, we utilize self-supervised machine learning approach to summarize the information of the SPOKE knowledge graph into numerical embeddings, where each numerical factor represents a node from the SPOKE graph. Then we feed this embedding into our model together with randomly selected initial gene pairs to observe and then to further train our model. Afterwards, we use an acquisition strategy to select the next batch of interactions to observe based on the predictions, which will be further used to fine-tune both the model and the embeddings. This process will repeat until the end of experimental budget. One of the key components of our method is graph convolution network, which we visualize in the middle bottom plot. One layer of the GNN computation corresponding to aggregation of information for each node from its neighbors in the graph, where we represent blue and red as different edge types. This captures the local topology information from the graph, and the order number of the neighbors we will aggregate over is defined by the number of layers in the network. This allows us to utilize the graph information in the most natural manner. Another key component of our method is our acquisition strategy, where we balance the exploration and exploitation by a linear annealing scheme, where at the first step, we select our entire 200 samples based on purely exploration scheme using the trace optimality condition from linear optimal experiment design. And slowly the scheme transition to a fully exploitation scheme at the final step, where we select the samples purely based on epsilon greedy scheme using the viral loads predictions. In the next section, we highlighted our main experimental results. The first plot is visualization of the percentage of the top 50 interactions that we're able to uncover across different steps. And the experiments are performed over 100 replicates to capture the uncertainty depends on the initial random selections. We compare our method with benchmark methods, including randomly sampling entries from the matrix, and then an approach where the initial batting is being fixed instead of being fine-tuned by the newly observed interaction turns. We showed that despite working with a large combinatorial space and only observe 10% of the entire matrix at the final step, we're able to uncover over 81% of the top 50 gene pairs in the end. In the next plot, we perform a pathway analysis, and a biological process enrichment plot shows the changing number of gene ontology biological processes terms over multiple runs of the active learning processes. Enrichment of terms related to transcription and translation are persistently being selected by the active learning model throughout the runs, indicating their significance in recognizing the HIV life cycles. And this is consistent with previous understanding of the biological processes. In conclusion, in this work, we demonstrate that by incorporating pre-existing knowledge graph into a deep active learning framework that adaptively updates the model and then embedding on the newly observed gene pairs is able to efficiently reduce the complexity of gene interaction experimental design. And then in our future work, we're interested in navigating more effective method to incorporate the large spoke database into our model and fine-tune our optimization acquisition strategy in the framework. Thank you for your time.